Hi, this is Pavithra Elumalai from Dr. Areejit Samal lab in the Computational Biology group of the Institute of Mathematical Sciences in Chennai, India. This video is a supporting information of the paper that you see on the screen. Demonstration of how we preprocessed functional MRI images into functional connectivity networks that we analyzed in our paper. Neuroimages are generally obtained by recruiting suitable subjects and scanning each subject for a period of time, acquiring two sets of scans, namely the structural MRI and the blood oxygen level dependent functional MRI. For our purpose, we acquired resting state functional MRI images of subjects with autism spectrum disorders and age-matched healthy controls from the Abide1 dataset. In this demo, we will show how we pre-process resting state fMRI images of two subjects from the raw image to the functional connectivity matrix. First, let us look at the raw data. I have created a folder on my desktop called CONN demo, which contains two subjects obtained from the Abide dataset. Each subject folder will contain two folders, namely the anatomical and the functional folder, and the anatomical folder contains the T1 weighted structural MRI file, and the functional folder contains the bold functional MRI images. Here for the subject we have 3 runs, but for uniformity we have used run 1 of every subject throughout the data set. Similarly, the other subject has the anatomical and functional folder, and the anatomical with T1 weighted and the functional with the bold images. Now, structural MRI images are three-dimensional images of the brain. They clearly have higher resolution compared to functional MRI images, and they help to differentiate between the different tissues in the brain. On the other hand, functional MRI images are four-dimensional images of the brain, meaning they are 3D images but over time. So every three-dimensional coordinate XYZ in the brain image has a time series associated with it for every value of the bold signal that this coordinate takes over the period of the scan acquisition time. So to pre-process these scans into networks, we use the MATLAB-based CONN toolbox. So now let us open CONN toolbox to set up the pre-processing. Open MATLAB and type CONN after installing CONN toolbox on your MATLAB. This will open the CONN GUI. So this is the CONN GUI, and let us open a new project. Please note that this GUI is different, looks different from the default dark theme of CONN, because I have changed it to a light theme for personal preference. So now I'm going to create a project on my desktop called CONN project demo. Click save, and this opens the blank project. Enter number of subjects, which is 2 in our case. Number of sessions is or runs is 1. Enter repetition time, which is 2.5 seconds for these two subjects, but this information is usually given out by the imaging site. Click on the structural tab on your left to enter or input structural MRI images into the project. Now on the box to your right, you will have to find the folder that contains the raw data, which in our case is the CONN demo on desktop. CONN demo on desktop, yeah. So we have found the two subject folders. Now we have to input the structural images and assign them to the subjects 1 and 2. So structural MRI usually have the extension t1w.nii.gz. Click find, which gives the two files for subject 1 and 2. So I'm assigning them to subject 1 and 2, and click import, which will import these files into the current project. Click OK. Now we can see that the images are assigned for subject 1 and 2. Similarly, click on the functional tab on the left to do the same for functional images. Now, on CONN demo, let us find the functional images, which usually have the extension bold.nii.gz. Click find, which gives all of the bold files. Since we are using only run 1, I'm assigning run 1 of subject 1 to subject 1. Click OK, and similarly for subject 2, run 1 of subject 2. Click import, which will import the files into the current project. Similarly, click OK. And now moving on to the ROIs tab on the left. Now I'm going to remove the default ROIs, which is Atlas and networks, which we are not interested in. Replacing that, we are going to enter a new file, Atlas file. We'll come to what is a Atlas file later, but now let's just input it. So I'm creating a new ROI called and naming it Schaefer, and uploading the Schaefer 200.nii file, which I have downloaded from the GitHub page of Schaefer Atlas. The link is given in the description, which is the Atlas file, and this file will be assigned for all the subjects in the project. And on tabs, conditions, covariates, and first level and second level covariates, we have not made any changes. So with that, setup is done, and let us move on to the actual pre-processing. Let us now recall that fMRI images are 3D images of the brain, which means that we have to pre-process them both spatially and temporally. Let us start with spatial pre-processing and begin with structural images. The first step is normalization. Since the subjects in the study may belong to different age groups and gender groups, their brains would differ in shape and size. So we normalize them to a template called Montreal Neurological Institute template, in MNI template in short, to reduce intersubject variability. So the next process is segmentation. The brain image is segmented into different brain tissues to generate gray matter, white matter, and cerebrospinal fluid masks, and we save all of this information for later. Now we move on to functional images. The first step in functional pre-processing is realignment. When a subject is being scanned in this MRI scanner, they tend to move their head. They basically can translate their head along the 3 axes or rotate their head along the 3 axes, causing 6 different types of motion. This might cause head movement related displacement in the acquired scan and may convey anomalous information regarding the pole signal acquired. So the purpose of this step is to realign the acquired images to the first scan of the first session as best as we could while estimating subject motion. The next step in the process is slice timing correction. Since fMRI images are acquired continuously and sequentially over time, functional data can be time shifted, and this step basically corrects any temporal misalignments. So the next step is outlier identification. In this step, we go through the functional scan and mark any acquisition whose frame-wise displacement is still greater than what we would like. So after marking this outliers, we save all of this information for later use. The next major step is to normalize the functional scans into the MNI space similar to structural volumes and to segment the functional volumes into different brain tissues. Now let us perform all of the steps on the CONN toolbox. So click on the Preprocessing tab on the left to preprocess. Now select the default preprocessing pipeline for volumetric analysis. Now it displays the steps in the default preprocessing pipeline, which has 11 steps by default. But since we're not going to be performing the last two steps, which is the functional smoothing in our method, I select them and click remove. Now we have 9 steps, 4 major steps for functional and 2 major steps for the structural, and I click start. Now CONN toolbox asks for the slice order of the functional scans. For our subjects, the slice order is interleaved signings. This information is usually given by the imaging site, and you can refer to the scan parameters given by them to find out what is the slice order for your data set. So with that, click OK. Next is the outlier detection preference. We are going to be using the conservative settings, which means we'll be removing acquisitions with frame-wise displacement greater than 0.5 mm. Now click OK. Yes, we'll be using the default tissue probability maps. Click OK. And this will start the pre-processing steps on the CONN toolbox, and the small pop-up window you're seeing will inform you about what current step is being performed by the CONN toolbox. So now note number 1. The steps and the parameters that we show in this video demonstrations are the steps and the parameters that we used in our paper, and please be advised that this is not the only way to do it. Note number 2. The pace at which CONN toolbox is performing steps in this video is not the actual pace. This video is like fastened to shorten the duration of the video, but in reality it takes around 15 minutes per subject, and if you have 2 subjects you can safely assume 30 minutes. But this estimate is also highly mission dependent. 15 minutes is on my laptop that I'm using. If you have powerful machines, the process could be fastened up, since CONN toolbox allows for processing all of these steps in multiple threads, meaning parallel computing. Now let us wait for CONN to finish all of the pre-processing steps. So now CONN has finished pre-processing, and let's click continue. Now to import all of the results of our pre-processing, we need to click the tab done on the left, and let us click start. So all the results are imported into our current project. This will import the results. With the appearance of the denoising window, which is the next window, we'll know spatial pre-processing of our dataset is done. Now After spatial preprocessing, we use information from structural and functional scans to obtain time series associated with every three-dimensional coordinate, also called the voxel. This is the information that we are interested in, and it will be a hero from this point onwards. Since this time series is the temporal component of our data, we will proceed with the temporal preprocessing or the denoising phase. Physiological effects such as heartbeat and metabolism can cause movements in the subject during scans, creating false spikes or fake spikes in the bold signals obtained, called the motion artifacts or noise, which we want to remove from our data. In the denoising phase, we use regression to remove any residual motion artifacts or noise from estimated subject motion, identified outliers, and the white matter and CSF areas, and we use the high pass filter to remove any low frequencies from the data. Now let us perform this process on the CONN toolbox. On the CONN denoising window, we can see the confounds for the linear regression step. So we can see 5 confounds from white matter and 5 from CSF. We can see 12 confounds from realignment, which is the 6 types of head motion and their first order derivatives. And the confounds that we see on the scrubbing tab are from the outliers that we identified from the previous step. Since we won't be using the effect of risk confound, we are going to be removing this confound by making the confound dimensions to be 0. Now we have to perform the high pass filtering by removing the low frequencies. To do that, under the band pass filter tab, set the frequencies to 0.008 to infinity to be included in our data. So as all the confounds are as we want them to be, and now we can click done. So yes, click start to start the denoising phase on the CONN toolbox. The small pop-up window is displaying what is the current status of the denoising on the CONN toolbox. Now let's wait for denoising to finish. While we are waiting, I reiterate that the parameters that we are using are the ones that we have used in the paper, and it is not the only parameters that you can use. With the appearance of this window, we know that the denoising phase on CONN is finished. After denoising, we would have removed noise and motion artifacts from the bold signals of every voxel as best as we could, and we can now start to analyze them. Now, these time series indicate the activations in the bold signals in every brain region, and any similarity or synchronization in this time series between distant regions of the brain is called functional connectivity. Since computing functional connectivity or similarities in time series between every pair of voxel can be time and compute intensive, we use parcellations. Parcels are obtained by grouping voxels, neighboring and related voxels, and we use the atlas file to guide us as to how to parcellate the brain. Recall that we input a atlas file during our setup phase called the Schaeffer 200 atlas, which parcellates the brain into 200 regions of interest. After parcellating the brain, we compute the average of the time series of all the voxels contained in a parcel to obtain a representative time series representing a region of interest. So since we used a Schaeffer 200 atlas, we obtained 200 time series for each of the 200 regions of interest. Now we compute the pairwise similarity between these 200 regions of interest by computing the pairwise Pearson's correlation, and when we repeat the same process for every region of interest, we would obtain a matrix called the functional connectivity matrix, which in our case is the resting state functional connectivity matrix. Now let us see how to obtain this matrix on the CONN toolbox. On the CONN first level analysis window, click on the ROI to ROI analysis tab on the left. Now, the analysis type is functional connectivity with ROI to ROI analysis. Now, analysis options are calculating bivariate correlation with no weighting. Let us click done and click start. This would open the second level analysis window on the CONN toolbox. Since we are not going to be performing any second level analysis on the CONN toolbox and we only want the graph out of it, we click on the graph theory tab to the left, and clicking it will compute the functional connectivity matrix for us, and it would open the graph theory window. This is the window where all the regions of interest are represented as nodes in the graph. We now need the network of all ROIs, which is what we have chosen in the network nodes tab. And we are interested in the correlation coefficient, so change cost to correlation coefficient. And since we need all the values from -1 to +1, I'm changing the threshold value to -1, so it will compute the complete graph of all the 200 values for all the 200 regions of interest from our parcellations. To get the functional connectivity matrix, click on export data, and now we can save this matrix anywhere we like. I am saving it on our CONN demo folder, saving it as ADJ underscore graph, and click save. So after we have saved the functional connectivity matrices, we can open MATLAB to look at the data that we saved. So now on MATLAB, load the file that we just saved from the graph theory window on CONN, which is the adj.networkmeasures.mat, and it will load as the results variable, which we can see in the workspace. Results is a structure, and the matrix that we want is the Z variable there. So it is 200 cross 200 for 200 regions and into 2 representing 2 subjects. So if we open Z, that's the matrix, but the Z is the z-scores of the correlation coefficient, and since we want the raw correlation coefficient, we can simply take tan h of this matrix to get the raw correlation coefficients. Yeah, that is our matrix. And if we want to visualize this functional connectivity matrix, we can use a histogram function. So I'm computing the histogram of the first subject. What we see is the functional connectivity distribution of the first subject in our analysis. Similarly, we can visualize the same thing for the second subject. The functional connectivity distribution, which takes a Gaussian value. And there we go, we have computed functional connectivity matrices from the scans. We have also utilized the CONN toolbox to generate QA reports, which we have used for our quality control in the paper. The QA report can be generated by clicking on the QA plots tab to the bottom left, which is available on both the pre-processing and the denoising stages. Clicking on it will create a QA report. We can name the QA report whatever we prefer, and click on create new plot, and we can select all the plots that we need for the report, and click start. This will generate all the plots and visualization we need. Since there are only 2 subjects in this demonstration, please refer to our paper for more details on how we use this visualizations for our quality control and exclusion criteria. Refer to our schematic for more details, and the link to our paper is provided in the description.